try to go through quickly enough to have some questions at the end. Otherwise, please feel encouraged to reach out and contact as we go, or contact me afterward at the molecular integrative oncology. So my emphasis is going to be the evidence-based targeting of cancer molecular networks with natural products, natural health product supplements, and repurposed pharmaceuticals. So let's break that down. Precision medicine, excuse me, plus integrative oncology. And I apologize, I'm right in the middle of cedar fever in Austin, Texas. Cedar fever is a big thing. We don't have ice snow very often. We have cedar pollen snow. So forgive me for clearing my throat, because that's where we are. Precision medicine plus integrative oncology. Well, let's look at what are each of those. Precision medicine is defined by the cancer.gov and the NIH as basically looking at the person's genes, proteins, and environment to prevent, diagnose, and treat disease. And that it's an emerging approach that focuses treatment on the particular and individuality, the variability that exists in each person. The basic criteria, the basic dogma of molecular biology is DNA makes RNA makes protein. Called DNA is transcribed into RNA, and RNA is translated into protein. But the thing to remember is DNA in the nucleus makes these bits of RNA, and RNA then goes out of the nucleus and then zip goes through a little bioprinter called a ribosome that makes protein. So DNA makes RNA makes protein. And that's what we're looking at, is that subject of those functions. DNA study is genomics, the RNA study is transcriptomics, and the protein study is proteomics. So that's where we're aiming. Whenever anyone talks about precision medicine, that's what they're talking about, is how to have medications aim at one or another or many of those components. Integrative oncology is a subset of cancer treatment which looks at the person's individual composition, and that using, in addition to conventional treatment, natural health product supplements and repurposed pharmaceuticals. In other words, pharmaceuticals that are already on the market, already approved by the FDA to be marketed, and that the physicians have access to, and that those drugs can be repurposed for other reasons other than what the FDA has approved them. And that's a very common practice in the USA, in Canada, and various other parts of the world. And that repurposing in oncology treatment, oncology doctors repurpose oncology drugs, so chemotherapy drugs and the targeted precision oncology biologic drugs, about 30% of the time. So it's a very common practice. What they don't do is repurpose the non-cancer drugs, where the researchers in cancer research have identified that non-cancer drugs like antibacterials, antifungals, non-steroidal anti-inflammatory drugs, proton pump inhibitors, statin drugs, a whole range, anti-diabetic drugs, a whole range of primary care pharmaceuticals that primary care doctors, family doctors, internal medicine doctors, other primary care doctors use, but that those can be repurposed for their anti-cancer molecular activity. And then if you combine these two, precision medicine and integrative oncology, that's where the field of molecular integrative oncology has been defined. That's where it's now arisen. So that's what we're looking at, is how do we take the evidence base that's available, and the evidence base means the scientific articles that have been published, and the ones that I focus on are the ones that are published through PubMed. PubMed is the main portal for the biomedical research that's indexed. Think of it as the electronic card catalog at the front of the library. It's not all the articles, it's not all the books, it's the electronic card catalog or the digital card catalog that gives access to where all those are around the world. And that right now there's about 32 million entries in that PubMed index. So everything that I'm talking about comes out through the PubMed portal. When you combine precision medicine and integrative oncology, that's the new field of molecular integrative oncology. And it uses the patient's particular data about any DNA, RNA, or protein data, plus the data that's in the evidence base about those particular cancer cell types, and then applies that using the evidence base that shows that natural products and repurposed pharmaceuticals have anti-cancer molecular effect in those cancer cell types and against those particular adverse mutations or alterations that are displayed. It's important to understand the big picture. There are common characteristics that exist in cancer called the hallmarks of cancer. And for about 20 years, we've looked at these 10 main characteristics. And if you look in the literature, go into the science articles about the hallmarks of cancer, what you'll see is this diagram. And I find it really a bit complex, a bit overwhelming. Let's break it down. Every one of these pictures, every one of these icons, these graphical icons, is associated with an associated characteristic called the hallmark, and that it's still complex. And so to make it a little more simple, what I've done is opened this graphic into a table, and then each one of those features are translated into a more user-friendly description. So DNA mutates and becomes unstable. There's an alteration in metabolism in the cancer cells that is very characteristic. And the cancer cells start growing, the signaling inside of them to grow is all the time on, and the ability to turn it off has been eliminated. And so they just keep growing. They resist the normal, automatic programmed recycling or the automatic programmed cell death. And there's relentless replication that then accumulates this lump of cells that then cultivate the growth of their own blood vessel supply to bring them oxygen, to bring them nutrients, to release and remove waste products. And that those cells also, the cancer cells, accumulate immune system cells that are used to feed and grow and protect the cancer cells from being identified and eliminated by the good anti-cancer component of the immune system. Therefore, the cancer lump or the tumor is able to avoid immune destruction. And along the way, at any point in time in this circuit of activity, the cancer cells can loosen their attachment to each other and release and go to another location that's called metastasis. But also, because they don't turn off their growing signals, they don't have that sensitivity to turn off the growth activity. When they hit a margin of an organ, they just keep going and can invade into another organ. So that basic overview of what's going on, you can see that in this sequential representation of how to understand the hallmarks. Just briefly went through that right now. I'm trying to give that overview of where we are in understanding cancer. In regular cancer treatment, the drugs are called targeted biologics. These are the new precision oncology drugs, or chemotherapy. And they can target each of the hallmarks of cancer. And here are some examples of the types of drugs that can target each of the hallmarks of cancer. Interestingly, when you look in the literature, natural product supplements, when you find particular ones, they can also target each of the hallmarks of cancer. And that repurposed primary care drugs, which are not approved by the FDA for marketing as anti-cancer drugs, so primary care pharmaceuticals also can target all of the hallmarks. So this opens up a very important set of options, a very important resource for anti-cancer treatment in addition to the conventional treatment. But how do we do it? We need to target the molecular pathways and molecular networks in cancer cells, also cancer stem cells. And because cancer cells recruit and induce the relocation of a bunch of different types of cells, blood vessel cells, immune system cells, a whole range of cells into the microscopic environment immediately adjacent to the cancer cells, those cells are called the tumor microenvironment cells. So we need to be able to target all of those and the molecular networks associated with it. So what I want to show here is a representation. This is the main cancer molecular network that is the most referenced, the most research citations. It's called the Kyoto Encyclopedia of Genes and Genomes, Pathways in Cancer Map. And if you look here, you could see some of the hallmarks that I was just describing, that the cancer cells make their own blood vessel supply. They avoid and are resisting automatic program cell death or automatic program, I call it automatic program cell death or automatic program cell recycling. And that's normal, healthy activity. And the cancer cells are avoiding that. Technically, it's called evading apoptosis, or the unstable DNA or the mutated DNA or that lack of ability to turn off the growing signals. And this is a map that shows a range of these hallmarks. It doesn't show all of them. We have to go to a bigger map to show the maps of the tumor microenvironment, for instance. And we'll notice that there are a number of pathways of activity. So molecules, each one of these indicates a molecule that's then communicating activity next to the next molecule. So this molecule signals this molecule, or this molecule signals this molecule. You'll see it in a cascade, a molecular cascade. The molecule gets immediately adjacent to the other molecule, causing a change in shape, a change in function. Sometimes they bind together, sometimes they break apart. When they change shape, that can cause a change in function. And then it goes to the next molecule and the next molecule into a sequence, a pathway. It's called a molecular pathway. And that here's a list of that same map showing the molecular pathways. And they have a bunch of unfamiliar type names. It doesn't matter what the names are right now. It's just to understand that these are the labels of those pathways, kind of like the labels on a highway. Shows you, okay, well, that's how you can identify that on a map and how you can navigate through that. Here's how you can navigate through these molecular pathways. And if we look right here at this blood vessel growth and the molecular pathway that's most identified with the induction or the creation of blood vessels for cancers, that we use this as an example, that the pathway is called VEGF pathway, VEGF signaling pathway. And that's associated with that hallmark of the developing the blood vessels. But then look here, there's more than one of these pathways that are coming into that characteristic, that feature, that development of blood vessels. And that identifies very importantly that even though the most common, the biggest main pathway associated with it is called VEGF, that VEGF pathway is not the only one. It's a network of pathways that contribute to that feature. And the dotted lines here indicate that there are multiple pathways underneath that. That's essentially showing you this version. If we were to zoom in more into the map, we would see that there's a whole range of additional pathways that participate in each one of these dotted line pathways. And so the point here is we're dealing with a molecular network on all of these hallmarks or all of these characteristics. And that by definition, we're having to understand and manage molecular networks in cancer cells, in cancer stem cells, and tumor microenvironment cells. Here's that map, the main pathways in cancer map with everything filled in at this high level view. But what do you do with that? How do you prioritize targeting? And that had a big boost in understanding about 4.5 years ago when the Pan-Cancer Atlas, which is an atlas, is a resource of maps, and the Pan-Cancer Atlas came out with a prioritization of pathways in order to identify, well, where do we aim? And that, what I'm going to show you is that after it was about a decade's activity of hundreds of scientists, that they looked at many thousands of cancer cells and looked for what are the alterations, what are the molecular changes that are associated with those cancer types, in order to be able to drive for researchers and for clinicians where to aim their treatments and where to combine treatments, how to combine treatments. And that was a great step forward. And here is that map. It's called the Flagship Paper Pathways of the Pan-Cancer Atlas. And it's still a little overwhelming, especially when you fill in all the molecular targets or the molecular addresses associated with it. So in order to make it easier for me to understand, I animated the map. So here's a particular molecular pathway associated with the feature called proliferation. So growing, dividing, accumulating, growing, dividing, accumulating, and cell survival and protein production. And this molecular cascade, this molecular sequencing of the signaling, every one of these molecular addresses or targets is a potential actionable alteration, meaning it can have an anti-cancer therapeutic option, that some drug or natural product can be used to target the molecular activity of that molecular site in an anti-cancer direction. And you'll notice in each one of these that there are features or characteristics identified in those pathways. And importantly, if I go fast forward through here, the feature which I think is most important, they're all connected. So this is a network of networks. And that's really important to understand, that because cancer cells are adaptable, they can compensate for adverse circumstances, just like healthy cells. So if you use a drug that targets a particular molecular site, the cancer cells can adapt their signaling around that. Think a main highway in which now a roadblock is put in and the traffic can work its way around it. That's what we're talking about in a molecular sense. And importantly, when you look at, I'm going to show 4 supplements, 4 natural product supplements that are very common. Curcumin from the root spice called turmeric, which is widely researched. Every yellow dot on this flagship paper pathway map, every yellow dot is where in the scientific literature, researchers have shown anti-cancer molecular effect at that molecular site. And so the idea here is curcumin can target any of these 44 different molecular sites that are identified on this map. Now, actually, there are many, many more molecular sites available that curcumin can also target, but on this highly selected map, the Pan-Cancer Atlas map, that this is the fundamental value of natural products is they are inherently multi-targeted. So the concept here is that if you could get 44 curcumin molecules into a cell, that theoretically you could target 44 different molecular interactions. And that's of great value because the cancer cells are adapting, and not all cells are identical because the DNA is mutated and unstable, that the progeny downstream grow, divide, accumulate cells, that they're not 100% identical to the upstream parental cells. And that's called tumor heterogeneity. You have groups of cells that are different. That means the molecular vulnerabilities in the cells in the cancer are different. And so being able to target multiple molecular sites with therapeutic agents is of great value. And then when the cancer cells are attempting to adapt, to work around an adverse change, so that now a drug that blocked a particular pathway, the cancer cells are now developing resistance to that by signaling around it. The natural products having multi-targeted activity can impede or inhibit or decrease the development of that resistance, another great value. The greatest limitation in natural products is they have a very low therapeutic index or very low therapeutic impact on their own by themselves. And so what the literature shows, and I'll demonstrate that going forward here, is that combining them in an overlapping synergistic way can provide therapeutic value greatly available at doses that are tolerable. So here's a next molecule, green tea extract. You'll see the green dots here, and there are 28 green dots throughout this flagship paper pathways map. And notice right here at this part, green tea, curcumin, green tea, curcumin, that the green tea and the curcumin can target the same molecular addresses in the same anti-cancer direction. And the idea here is that and it's demonstrated in the literature that curcumin can be synergistic with green tea. And when synergy occurs, the amount of each of the therapeutic agents required to get the same effect can be drastically reduced, can be reduced more than by half, can be reduced by 10 times, can even be reduced by 100 times. So 1 to 2 orders of magnitude value by overlapping the therapeutic agents. Look down here, the curcumin targeting these 2 addresses or molecular sites, and the green tea targeting these 2. And so combining them, now we get more molecular network targeting plus the synergy. So there's great value in looking at the literature of how to combine these. If we look at 2 more, genistein from soybeans, the pink dots here. Genistein is so widely researched that for decades, it was a standard in the cancer research literature, compared with which new items were compared. And then resveratrol, grape extract, or from Japanese knotweed and others. Most importantly, when we combine these 4 items, look here at this map. Everywhere you see multicolored dots is an opportunity for overlapping therapeutic anti-cancer synergy, evidence-based, molecularly network targeted. And that this is the great value of natural products, is combining them. If I show the repurposed pharmaceuticals, which we don't have time for, it can also add to this. And so the same information being shown here in the column. The first column is all those targets. Everywhere you see a row that has more than 1 check mark is an opportunity for overlapping therapeutic anti-cancer synergy. this all became validated in the research literature in the December of 2015 in a very high impact mainstream conventional cancer journal that put out a special issue about a broad spectrum design for cancer treatment and prevention. And that they looked at each one of the hallmarks. And what I'm showing you here is the articles, each one of the hallmarks. And there was a group of scientists that did this project. It took them 3 years to do this project. And in that project, what they did was look at all the hallmarks separately. And of those groups of scientists looking at various hallmarks, they came to the conclusion that it's a rational approach to use a broad spectrum of natural products and repurposed pharmaceuticals in addition to conventional oncology treatment. And a group came from that research affiliation that's now seeking to do clinical trials, and that looking at how do you apply a broad spectrum of natural products and repurposed pharmaceuticals. And I was recruited onto that board, and we have gone to the FDA. The FDA has given notification that they would give us waivers because we're not trying to get any particular drug approved to be marketed. Instead, we're looking at, well, how can we compare people who are getting all best conventional treatment to people getting all best conventional treatment plus the molecular integrative oncology in addition to their all best conventional treatment. And I've been doing this with my own patients since the late 1990s, early 2000s. And so that's why I got recruited onto this board. We have gotten no funding because the idea of doing the comparison without trying to find a new drug to be approved, hasn't been any backers. So if anybody is interested or knows some organization that would be supportive for research, then please contact me. So the final slide here is that there are benefits for natural products and repurposed pharmaceuticals. We could target the molecular networks, synergistic anti-cancer activity. It's rational, it is in the evidence base. It has a relatively high margin of safety. Nothing is zero risk, but it's a very manageable risk, and that these items are immediately available, that patients don't have to wait for years for the research to be brought to market. There are limitations, the low absorbability, the low therapeutic impact, and that these are not standardized out on the market, and people have to go and get all of these things themselves, and that because these are not patentable molecules, we don't have clinical trials. So I appreciate your time and the opportunity to share this information. If you have any questions, please feel free to reach out to me, either directly by phone to my office, or you can go through the website, LaVallyMDprotocols.com. I'm looking at the questions.
Dr. LaVally, thank you so much.